Ladies and gentlemen, good afternoon. It's a great pleasure to be here to share with some of our work, some of our thoughts about the future of computational cardiac dynamics. So my name is Hengwei Zhang from Beijing Academy of Artificial Intelligence and also from the University of Manchester. So I will organize this presentation in the following way. First of all, I'd like to give you some background introduction. We know we have a lot of kind of challenges in cardiology. One of these grand challenges is about how do we understand the initiation and the treatment of cardiac arrhythmia. So especially with the kind of a regular heartbeat, more or less regular heartbeat, developed into suddenly this kind of a chaotic reason. Why it's happened, how it's happened, and what if we do something? Can we predict this kind of unsetting of this cardiac arrhythmia? So this is the golden three-chain questions is what attract most of kind of research studies. There's two ways of this kind of research. One way is a classical kind of approach called reductionist. What we do normally is we have the heart, then we use the scissor, chop into pieces to get this kind of anatomical structure to know what would be the chambers structure, what would be the bundles structure. And we also can get this detailed about the tissue histology, about the cell organization, fiber orientation, and etc. We can do further to get this kind of cellular information, especially about the cell memory. What about this protein structure, ion channels, so on and so forth. We can even go further to get this molecular basis of these cardiac functions, such as kind of expression of proteins, the genetic sequence. So this is a normal way we do for cardiac research, but there's a problem with this. The further the way we go down from this organ to this detailed molecular level, the far we are away from the functions. So the question here, what would be the logic link between this molecular information and the functions of the heart? Because we know the structure determines the function, but such a determination is not a one-to-one mapping. Okay, so in the way, we are missing a cross bridge, link the information from this molecular level to the organ level. So there's a second approach, is what we call the integrative approach. So this approach actually is a non-human dream, trying to say whether or not we can use a mathematical way to describe the functions of a biological system, such as the heart. So this is a non-dream, but fortunately, we have the work done by Hodgkin Huxley, who laid down the ground foundation for this type of research. In their pioneering work that developed a mathematical model for simulating the electrical action potential of the neurons in the 1950s. Because of this, they won Nobel Prize. After this, many works have been done in the cardiac field, try to develop similar mathematical equations, try to predict the functions of the heart. Especially recently, we know AI helps the scientific research. So AI for science is a hot research topic at the moment. For AI for science, actually, it can facilitate or helps for cardiac researchers. At the moment, there's two parallel ways for AI application in cardiac research. One way is what we call data-driven. Okay, so then we treat this kind of heart as a black box. We use multimodality data, such as this text language data, the image data, and also molecular data, structural data. So in the way, we put all this modality kind of data into this black box. We try to develop an algorithm to predict and diagnose cardiac disease. But there's a question here. The question here, we can do the prediction, but the prediction is unexplainable. So there's another way, is what we call the physics-driven. Or in other words, we treat the heart as a white box. We use the similar kind of data, the same source of data, but we use the principles of physiology, principles of physics, and mathematics. So in the way, we can develop another set of models. In the way, we can not only diagnose, predict the cardiac disease, but also try to understand what are the underlying mechanisms. So in the way, we try to say, if we understand the mechanism better, we hope we can have a better treatment. So at the moment, we are working with Professor Lee together here. Okay, so we try to combine these two approaches together. Try to say whether we can combine both the data-driven and also principle-driven together. So in the way to develop a digital twin of the heart. So this is kind of a foot plan for this kind of protocol for the research. So we have a multimodal data from text, such as the electrical record, and to the structural language, such as this image omics, and also functional language, such as ECG. Okay, so then we combine all this together from clinical and also from the bank badge. Okay, web science, such as page clamp. So in the way, we put this data into kind of a deep learning model, try to do these predictions. In other way, we combine this data together to develop the model from ion channel to cell to tissue to the whole organ, even to the whole human heart. We combine them together for personalized model development. So in this way, we can have a digital twin of the heart. With the hope, digital twin of the heart can help us to do better for precise medicine. So first of all, I'd like to introduce about this kind of principle-driven of this work. Okay, so this is what we've done for the virtual heart development. For the virtual heart development, we need many details. The details including the structure, include these electrophysiological properties. So in the way, we can put the electrical information about the structured information together to form a platform. So in the way, we can simulate the electrical and the mechanical dynamics of the heart, and also the fluid dynamics as well. So in this way, we put them together. We hope we can develop a model for the heart. For imaging-wise, we use a different modality to get the detailed structure of the heart. From kind of a visible human project, then we develop an AI algorithm to do this image processing for the segmentation and then 3D reconstruction. You can see here, we can reconstruct the 3D whole heart model for the human, different color to denote the different special regions of the heart, including the atrial, ventricle, and the Purkinje fiber network, and so on. But this kind of image lack information about the fiber orientations, because the fiber orientation play important role for cardiac excitation conduction, somehow like a high speed kind of a pathway for the electrical conduction. So for cardiac modeling, we need such important information. To get this information, what we did use here, use a diffusion tensor MRI. So in a way, we can get not only the structure, but also this cell arrangement and the fiber bundles in the tissue. But these two methods are really time-consuming. So now we develop another way which gives us a high, fast, and efficient way. Try to use the X-ray, this micro-CT, to do the soft tissue. We know X-ray is transparent to soft tissue. What we did here, we soak this hard tissue into iodine. So in the way, we can turn this soft tissue to be visible by X-ray. So in the way, we can have a fast and high spatial resolution imaging. So the spatial resolution we got there can be 10 micrometers. At even some focus region, we can get 1 or 2 micrometers. So this is a very high spatial resolution images. Then come back to the electrophysiological model. We developed a model for these cardiac systems, not only for different regions of the heart, for different cell types, but also consider this kind of activation and inactivation of these ion channels. because for these different cells got the different ion channels that work with the relatively kind of different in the kinetics and also the current densities. So all these kind of ion channels work together to produce these ion action potentials. So we put all this electrophysiological data from patch clamp together, so we can use a mathematical way to describe the cell for cell memory ion channels and also the intracellular ion transportation and the circulation. Okay, so in the way we can put all this together, so from this single ion channel to the cell, to the tissue, then to the whole organ. Then now we are going even one step further, trying to bring this molecular dynamic structure, such as this protein structure, into this kind of a simulation to see how the altered structure would alter the function of the heart. So this is kind of a hierarchical models ranging from the ion channel to the cell, to the tissue, to the whole organ. So this slide shows you the simulate initiation and the conduction of electrical exciting waves in the whole heart. What we got here, different regions, we got different cell model. So this is including the synaptic node, the atrioventricular node, Purkinje fiber, and also this endo-medial epi layer of the ventricles. So together, we can have over 27 different cell types. Each of them have intrinsic differences in their electrophysiology. So after we've done the work, we needed to validate the model. How do we know the model have this physiological relevance? So to test this one, what we did here to simulate the actions of acetylcholine when vagatine is active. So acetylcholine normally slow down this heart rate and also can cause changes in this electrical excitation wave conductions. So this would be reflected by the change in the RI interval, change in the PR interval, change in the QT interval. So this slide here shows the simulated ECGs for the control and for the application of acetylcholine. So this is just a simulation. So then we have the experimental data to compare the simulation data. So in the way, you can see the top panel is simulation, the bottom panel is experiments. Both quantitatively and quantitatively, they are similar. So in this way, this validates the work or validates the relevance of the model developments. So we can go further, not only just the single lead ECG, we can simulate the multi-lead ECG here. So what we've done here, we bring the whole heart model into the human torso. In the torso, we consider the bone, the lung, and the stomach structures, the soft tissues, and also heart tissues as well. So in the way, we can put the heart into the tissue here, then we can put the electrodes as you normally do at the clinical settings. So in this way, we can simulate not only 12 lead ECG, but multi-lead ECG arrays. Here, to show you here, so the 36 lead ECG put on the front chest. Okay, the red line here demonstrated experimental recordings. The blue line here shows the simulation. Okay, in the control case here, you can see this ECG in all 36 of these ECGs resemble to the clinical recordings. And also we can simulate this kind of conditions with ischemia, which can cause changes in the ST segment. And also in some leads, you can see the change in the T waves. Not only the ECG, but also the MCG, which is magnetic cardiogram. I believe MCG would be much more popular in 10 years' time for clinical diagnosis because ECG decay in the tissue, but MCG doesn't. For some small ischemia region, ECG may not be sensible, but the MCG could be a powerful tool. Okay, so this is just the hope in the way the MCG would play most important role. Not only just the electrical, but also we can simulate the electrical and the mechanical coupling. Here to show the human atria. Okay, in the left panel here is a normal tissue. In the right panel here is the atria with atrial fibrillation. You can see multiple wavelets there. So this multiple needs desynchronize the mechanical contraction. So in the way, the atrial lost the contraction power. Okay. So then the next thing here, how can we put all these things, data-driven, principle-driven together to develop the digital twin of the heart? For the digital twin of the heart, we needed to have this kind of a structural twin. So in the way, we can have these images, clinical images from a different modality. So in the way, we can have clinical images, we can use AI to segment this tissue structure of the heart, and also this ischemia scar regions as well. Okay, so then we can combine this structural, the scar, and this fiber bundles together. Then we can also lead the functional chain. The functional chain starts from these electrical action potentials. And then in a way, we can put them together, we can simulate the ECG compared to the personal ECG. So there may be a difference between these two. Then we can use AI algorithm to optimize the parameters in our electrical model. So in the way to let this the whole heart model can resemble this personalized human heart functions. So the structure is from this imaging. So they are accurate. Only thing here is kind of can be played a little is this kind of electrical. So we use AI algorithm to do it. So to show here, not only just the 3D, we can also do the four-dimensional reconstruction from the clinical images. Okay, so in the way, we can have this contraction. This contraction can help us to do some biomechanical contractions to develop the kind of a special distribution of the strain and the stress of this cardiac muscle. Because at the moment, we use the ejection fraction for heart failure kind of diagnosis, but ejection fraction may not be sufficient. We hope use this work, we can have this atlas of the strain and the stress. Okay, so then we put them together. So in the way, from an image, from this kind of ECG measurement, we put them together. We hope we can have a personalized the human heart and the human torso. Okay, so then the potential applications here. To summarize, we got this multi-scale computer models of the heart, ranging from the ion channel to the cell, to the tissue, to the whole organ. And also in the lab here, we also develop 256 leads ECG vest. We can measure this ECG from the front and the back of the human torso. So then we combine together. So then in a way, we hope we can do some applications. First application is use the virtual heart and this 256 ECG for this kind of electrical function imaging of the heart to pin down this kind of atrial fibrillation orange. So in a way to have this precise kind of AF ablation. And also we can test the drugs which can be anti-arrhythmic or whether they are not have their kind of cardiac toxicity. And also we can do this virtual surgery planning. Okay, so this is something, because of time, I only show you the way we did for this kind of application. What we did here, we use the kind of images, then we reconstruct the human atria. Okay, then we put this structured information with electrophysiological model. We can see what kind of a protocol would be more efficient for this ablation of atrial fibrillation. What we said here, we can have this kind of normal sinus excitation, but then we can have this kind of HF ablation. Then we can have when protocol ablation doesn't work, then we can have a T-type, then in a way we can get rid of this kind of atrial fibrillation. So this is just to show you the way we can do. We can have this platform for drug test as well. And also we can do this kind of cardiac imaging. For the cardiac imaging here, what we found, we need not only just the 256 ECG, we need at least over 1,000 electric leads put on the chest. So this calls for further research in the material. So in the way to kind of have a variable, comfortable kind of a device for such kind of a ECG kind of measurement. Okay, so this work being carried on for a while now, but still there are many challenges. One of the challenges here is a larger scale of the computing, especially if we wanted to put this into clinical application, the simulation time is a bigger bottleneck. So last year, we developed a kind of a parallel computing system which enables us to do the real-time simulation, which means 1 second of the hard time, we can use 1 second of computing time to do these functions. Okay. Looking into the future, we hope of starting from here, we can develop this not only just the heart and the circulation systems, and also other important organs in the kind of body. Hope we can, from here, to develop a digital life. Thank you very much for your attention.